What's the pain with 2021 number 1, membrane transport, cell signaling, and inhibition. Polycystic kidney disease, an inherited disease that causes water loss from the body and affects cell division in the kidneys. Because water movement across cell membranes is related to ion movement, scientists investigate sodium potassium ATPase, known as the sodium potassium pump, in this disease. The albumin, a steroid hormone, binds the sodium potassium pump in the plasma membranes. Individuals with PKD have a genetic mutation that results in increased binding of albumin to the sodium potassium pump. The scientists treated normal kidney cells and PKD cells with increasing concentration of albumin and measured the number of cells and the activity of the ATPase. After a period of time, the scientists hypothesized as a cell transduction pathway that includes the protein kinases MEK and ERK may play a role in PKD symptoms. Wow, that is a lot of alphabet soup. So we see that the albumin, of course, binds to the sodium potassium pump. Individuals with PKD, of course, have that increased binding. Those are important things for us to know. So as you're going through questions, underline the important stuff that you're going to need to know for later. They give us these 3 different diagrams. They give us a graph showing us the number of cells. They give us the sodium potassium pump activity, and then they also give us the pathway showing that albumin in the sodium potassium pump with the signal transduction for the transcription of the genes. So describe the characteristic of plasma membrane that prevents simple diffusion of sodium and potassium across the membrane. So as always, A has to do with the biology question. Why is sodium, which is a positively charged ion, and potassium, which is also a cation, unable to get across the membrane? It's because they're charged ions. The plasma membrane is hydrophobic, and so the interior of the membrane has those hydrophobic tails, and then you have your hydrophilic heads. And so the membrane itself is hydrophobic, which inhibits a charged molecule from passing the membrane. So the interior is hydrophobic, the phospholipid tails are hydrophobic, the exterior is hydrophilic, and the hydrophilic heads are, of course, hydrophilic or polar. And then you have to explain why ATP is required for the activity of the sodium potassium pump. Well, since we're talking about a pump, we know that something's going against this concentration gradient. So sodium potassium pumps are pumping ions against their concentration gradient, and this requires some type of input of energy, which would be that ATP that is used. So student says plasma membrane is composed of phospholipids, which have a hydrophilic phosphate head and inward-facing hydrophobic fatty acid tails. But the plasma membrane is semi-permeable, meaning that only nonpolar small substances undergo simple diffusion through this membrane. Since sodium potassium ions, they have an electric charge, they must enter the cell through transport proteins since can't diffuse through the membrane. Because cells create a gradient of potassium ions within the cell, ATP is required to transport sodium potassium across the pump. Since the cell is bringing in more potassium ions against their concentration gradient and pumping sodium against their concentration gradient, energy in the form of ATP is necessary for this protein to work. So B says, identify the dependent variable in the experiment represented in figure 1. So the dependent variable is what is being measured, right? So the scientist has changed something, which is your independent variable, and you're looking at what is being measured. What's the result of that change? And so that's traditionally found on your y-axis. So the number of cells is what is being measured. They're seeing how many cells are going to be made in those different levels, just by the use of the normal human kidney cells as a control in the experiment. So we want to see, what does PKD do to the cells? Okay, so having that increased binding to albumin, how does that actually affect the number of cells? And so we need to have the NHKs, the normal cells, in order to compare. So allow scientists to determine the effect of PKD on the cell's responses to those different levels of albumin, and allow scientists to compare the responses of PKD cells and normal cells to that albumin. So next we have to justify the use of the range. So why do I have a multiple range of concentrations? Well, because I don't know whether different concentrations are going to have different effects on the cells, and they didn't know which concentration there would be an effect, or what they're looking for here. So dependent variable is the number of resulting cells of NHK and PKD following the different concentrations of albumin. NHK cells serve as a model to compare the cellular changes caused by the albumin and PKD cells, since the values collected would be insignificant without a relevant comparison. Using a range of albumin concentration allowed research to observe changes that occur in cells in multiple conditions, since a very small amount of albumin may have a very different effect on kidney cells compared to a very high amount. So C says, describe the relationship between the concentration of albumin and the sodium potassium pumps activity in both types of cells. And so if you look here, you can see that there is a decrease in the activity as there's an increase in the albumin. So as albumin increases, activity is going to decrease, which is an inverse effect. So increasing concentrations results in decreasing activity. There is an inverse relationship, negative correlation between the concentration. So next I have to do is the scientists determined that sodium potassium activity treated with 1 picomole molar in 150 units of ATP hydrolyzed per second. Calculate the expected activity in PKD cells treated with 10.6 albumin. So if I go 10 to the 6 right here, I can see that there's a 30% activity, okay? So if there's 150, 30% of 150 is just going to be 45. So the answer was somewhere between 40 and 50. They gave a range depending on how you read the data table, like how you read the chart. If you read a certain way, it may have ended up being 40 or may have been 50. I personally just read it as 45. So based on the data shown in figure 2, describe the relationship between, this is what we already did. In both normal cells, we see that the concentration increases past the 10 to the 4th, a drastic decrease occurs. So there's a negative relationship between the albumin and the ATP activity. In PKD cells treated with 10 to the 6, we see that the activity is 45 units of ATP hydrolyzed per second. So in a third experiment, scientists added an inhibitor, which is phosphorylated MEK, which is PMEK, to the PKD cells exposed to 10 to the 4th albumin. Based on the figure, predict the change in the relative ratio of ERK to PERK in albumin-treated PKD cells with the inhibitor compared with albumin-treated PKD cells without the inhibitor. Provide reason to justify the prediction. And so here I can see that if this part doesn't take place, right? Then this is not going to allow for the phosphorylation of ERK to PERK. And so because of the fact that there is a decrease in this, we're going to find that the ratio of the ERK to PERK is going to increase because we're not going to be converting it into PERK. Now you could have said that the ratio would stay the same with the inhibitor. That is another option. And so we can see that the reasoning why. So you have to justify. The amount of PERK would not increase as it does without the inhibitor. The amount of PERK will not decrease as it does without the inhibitor. The cell continues to synthesize ERK, but you see we're not phosphorylating it. And then the phosphorylation of ERK to being phosphorylated. Okay. So we're seeing that there's a lot taking place there. You could have said the option 2, which was talking about that no ERK was synthesized and that PERK was not being dephosphorylated, because that was also an option. So as you said, relative ratio will increase in cells treated with the inhibitor compared to cells not treated with the inhibitor. Since PMEK signals the transfer of ERK to PERK, if this PMEK is inhibited, it won't allow for the phosphorylation of ERK to PERK, and the amount of ERK will increase relative to PERK, causing an increase in ratio. As you see, still alphabet soup. So using data in figure 1 and the signal transduction pathway represented in figure 3, explain how concentration of cyclin proteins may increase in PKD cells treated with 10 to the 4th albumin. So here you can see a 10 to the 4th albumin. We have this large increase in PKD cells. It's the highest that we see. And so if we follow the pathway through, we can see that that albumin, of course, causes the increase of PMEK, which then causes PERK, which then is going to act as a transcription factor and increase the transcription of these target genes. These target genes are going to make more cyclin. So at the cell number increases at 10 to the 4th because it stimulates transcription of cyclin, and cyclin is needed to regulate the cell cycle. So albumin is a signaling molecule that causes transcription of target genes in kidney cells. Since PKD cells have increased binding of albumin, the cells in the concentration of 10 to the 4th have significantly higher levels of cells. It can be inferred that albumin increases transcription of the cyclin genes, which promotes cell division. Hope that was helpful. Remember, 8 biochemistry is success. Bye, y'all.